I'll wait just a little. We'll see how it goes. Okay, today is part 2 of population genetics at the microevolutionary scale. Migration, drift, and non-random mating is what we're talking about. So this is just to quickly refresh you from the long weekend. Hardy-Weinberg equilibrium conditions apply if there's no mutation, no selection, no migration, genetic drift, and no non-random matings. Obviously, none of those things really apply, but if they were to, then you would have no change in allele frequency in populations over time. You'd be able to describe the frequencies of alleles if there's just 2. P is the frequency of one allele, Q is the frequency of the alternative, and the frequencies of both of them add up to 100%, right? And obviously this applies if there's lots and lots of alleles as well. The genotype frequencies, you can calculate the expected genotype frequencies from as p squared plus 2pq plus q squared equals 1. It's all ringing a bell from last week. We didn't lose too much then. Good. So, of these 5 things that you can, in a evolutionary, sort of microevolutionary population genetic sense, say these 5 things are what cause allele frequency change. We went through 2, mutation and selection last time. Migration is the one where, or one of a couple where the terminology confuses people a little bit, because we're again have to be thinking at the gene level, not at the organism level. So no migration is not in reference to organisms migrating seasonally or organisms moving in and out of a population directly, although it's related to that. What we're really talking about is whether alleles move in and out of populations at some rate that is different from what you would expect based on their frequencies in those populations. In other words, if there's biased migration, if certain genotypes move into or out of populations more than you would expect by chance. If they do, we call this gene flow. So gene flow is, in a sense, the movement of alleles across the landscape from one population to the next. So it could be organisms flying or moving from one population to the next, carrying alleles that are different from the ones that are in the population they're going into, or at a different frequency than the population that they're going into. It could also be things like pollen moving into a different plant community, which is the haploid state. What gene flow does, and it's going to become really important when we talk about speciation later, what gene flow does is it makes different populations more similar to each other genetically. So when there's movement of alleles between populations, it has a homogenizing effect, so that they become less genetically distinct. When we get to speciation, obviously the hallmark of speciation is that there's not gene flow between these populations, and then they have independent evolutionary trajectories, and we call them species. So file that away until speciation later. So here's this kind of silly little mouse wheel of alleles in a population. We're starting with a population that is fixed for this A1 allele. That allele is at 100%. So when those gametes combine to make genotypes, you only get the A1A1 homozygous genotype. But then what happens is a bunch of individuals from a different population migrate in, bringing with them alleles that are different from what's present. In this extreme example, bringing in a completely novel allele that wasn't present in the population before. They bring in the A2 allele. So these then become the genotype frequencies in the interbreeding population that makes gametes that has allele frequencies that are different from what was there before. That ought to be super obvious and easy to see. Now, what's less obvious is that it's the exact same thing if, suppose, this initial allele frequencies were 50-50 and there was migration of individuals that had brought in the same alleles but at frequency of 80-20. That would also change the allele frequency of the final population. So there's this kind of cool example in the book about these snakes that live on islands in the Great Lakes, this Nerodia cypedon. And hopefully what you can see in this picture is that the snakes vary in their color patterns. These are offshore islands out in the Great Lakes. And on the mainland, there's selection for banded snakes, because on the mainland there's trees, and so there's dappled shade and sun, and the snakes blend in and are less visible to predators if they have this kind of banded pattern. But these islands are sort of glacial remnants and don't have trees. They're these barren limestone surfaces. So snakes that are up on these rocks basking in the sun, if they were banded, they would be very conspicuous to predators such as hawks. So having no bands is selectively advantageous on these islands. So now, if you think back to the selection lecture, I said, well, what's the equilibrium frequency of a beneficial allele? I said, well, if selection is the only thing happening, it's 100%. So you would imagine, perhaps, these islands are favoring the unbanded alleles. And yet there's still banded snakes there, meaning there's still banded alleles there. And the reason why is that selection on these islands is eliminating the banded alleles, except new snakes from the mainland keep showing up, bringing with them banded alleles. So there's some kind of non-zero frequency of the banded alleles on these islands due to a balance between selection getting rid of banded alleles and migration or gene flow bringing these banded alleles back onto these islands. So you could picture a migration selection balance somewhat analogously to a mutation selection balance that we talked about the other day. So as these alleles move into these populations of island snakes, that's obviously changing the allele frequencies. It's making the island population more similar to the mainland population genetically, even though that's not advantageous. You could have snakes go from the island to the mainland carrying unbanded alleles with them. It would have a trivial effect, though, because the mainland populations are huge and the island populations are really small. So that there would be much less of an appreciable change in allele frequency if snakes went the other way, which presumably they do. But then on the mainland, there's selection against the unbanded alleles that come from the islands. So I just said that migration or gene flow makes populations more similar. Obviously, we'd want some way to quantify the genetic similarity of populations. And the most common statistic for doing this is called FST. It's a test statistic that developed from analysis of variance. It ranges from 0 to 1. And what it's doing is it's saying how much of the total variation is due to variation within subpopulations, there's differences among these populations relative to the total level of variation. That's what the S and T are for. And if FST is a high number, meaning maybe above 0.2 or something like that, then the populations are pretty distinct. If FST was 1, they would be genetically completely independent kinds of populations. If FST is low, the subpopulations are very intermixed. So if you measured the FST within a single population, you would have no subpopulations. It would be completely interbreeding one homogeneous population, and then in sort of theory, the FST would be 0 in that case. In practice, that doesn't really happen. So there's this other example in the book to illustrate FSTs and how they change over time. It's kind of a nice transition to some of the other stuff we're talking about. This is a plant called Silene dioica, and it lives on Swedish islands off the mainland of Sweden. And what happens is these plants colonize young islands. So these islands have been glaciated and they're rebounding and ecologically changing over time through ecological succession. So the early colonizers of these more barren islands are seeds of Silene dioica. And when seeds get to these different islands and start to grow into plants, the genes that come with those seeds are some sort of haphazardly random sample of what was in the larger mainland population. They're not necessarily representative of the genetics of the whole population because only a few seeds make it to any particular island. Then they grow up, they're pollinated by insects, and you get these little subpopulations of these plants on these different islands. But insects that pollinate these plants can fly around island to island, and when they do, they take pollen with them. So over time, the island subpopulations become genetically more similar. These insects moving pollen around are agents of gene flow between populations. And then, as the islands are getting older and older, and you get more shrubs and trees and stuff like that, the Silene dioica populations die off, and as they die off, as they're contracting, the subset of alleles that's left over time becomes once again a sort of non-random sample of what was there before. So that you would expect then the islands to become more different from each other through the extinction of and loss of particular alleles over time. So that's our kind of prediction. Here's FST values for young, intermediate, and old populations. And you can see, I hope, that even though these FST numbers are all really low, 0.03, 0.06, that they're higher when young, and the intermediate ones have the lowest FST values. And this change here is due to gene flow. It's due to these insect pollinators moving alleles between these different islands, and again, it makes them more similar. So this is kind of the nice example just because it transitions us to the next topic in this, which is genetic drift. In their study of these plants, the high or higher FST values for the young initial populations are due to random sampling error from this big population that is on the Swedish mainland. So as these seeds initially colonize these islands, which alleles get there is basically a matter of chance. So the likelihood of the same ones being representative of the whole population on each of these different islands is pretty small. That error, in a sense, due to sampling variance in small populations is what is called genetic drift. So students in the past have sometimes in their minds confused genetic drift and gene flow because drift, sort of, you can picture genes drifting across a landscape on the wind or something like that. Don't do that. They're just completely different sorts of ideas. Gene flow is movement of alleles among populations, making them more similar to each other. Genetic drift is random sampling variance within a population, and that can cause that population to differ genetically over time. So you have to sort of appreciate how this happens, you have to adopt a little bit of a statistical or at least a probabilistic kind of way of thinking about events. And one of the easier ways to do this is think about flipping a coin. We're going to imagine it's a fair coin. So there's 50% chance of getting heads and 50% chance of getting tails, right? If you flipped that an infinite number of times, you ought to get really pretty close to 50-50. Do you think if you flipped it 10 million times, you'd be probably about 5 million heads and about 5 million tails? Would you have exactly 5 million heads and exactly 5 million tails? That's not very likely. Now, at the other end of the extreme, if you flip a coin 4 times, you have a really significant chance of getting either all heads or all tails, 12.5%, just by random chance. If you flip a coin once, what's the chance of getting half heads and half tails? 0. You can only get a head or a tail, right? Or it can land right on the edge and stay there. So, just as an effect of sampling error in small groups, taking small samples, you're likely to deviate from the true null expectation of 50-50. And the same thing happens with gametes forming zygotes, and other random processes can cause departures from your expected allele frequencies just by chance, but they're much greater effects in small populations because of the statistical sampling involved. So if you have random departures that are averaged over a huge sample size, their net effect is usually very small. But random departures averaged over a small sample size can be significantly different from what you'd expect based on random chance. So here's this population of, or gene pool of mouse gametes. And if you have initial allele frequencies of 60-40, you, under Hardy-Weinberg, you expect the homozygous A1A1 genotype to be produced at what frequency? 0.36, 36%. If you look at this population of zygotes, what's the frequency of that A1A1 genotype? There's, in case you can't see it, there's 6 of them, 2 heterozygotes and 2 of the other type of homozygote. There's 6 out of 10 are the A1A1 genotype, so that's 60%. Your expectation is 36%, and you have a lot more of these produced, just by chance, as these gametes combine. If you went on making 10 million zygotes, you would expect to approach much closer to that 36% Hardy-Weinberg proportion. So anyway, if they grow up with no selection, nothing changes, and then they make gametes, the gene pool's different than it was before. You now have 70-30 allele frequencies just by chance. There's no selection involved. This doesn't imply one allele is better than the other, anything like that. Just because you have a small statistical sample, these chance events have magnified kinds of effects. So, this is a statistical graph of probability for this particular example. So if you had an allele frequency of 60%, like in that previous example, what's the likelihood of getting no change if you only make 10 zygotes? It's kind of like, what's the likelihood of getting 50-50 heads and tails if you only flip the coin once, twice, or a few times? You only have about an 18% chance of getting no change in allele frequency if you only make 10 zygotes. If you have a small sample, it's likely to be significantly different from what you had before. You have, a little bit over 80% chance of the allele frequencies being different than they were before, just because of statistically random effects. So here's kind of where it gets more interesting and important is, how big a deal is this evolutionarily? And the answer is it depends an awful lot on this sample size because of this statistical averaging. So here's a simulation model that plots the cumulative number of zygotes made. So here's the low numbers, like the 10 that we just looked at, and here's out 200, 300, whatever that is, 250. And what you see is, because these effects are random, you don't get the same trajectory of the model each time. The first time it starts out low, second time starts out high. It bounces up and down quite a lot with the small sample size, but as you start making lots and lots and lots of these things, what do they all converge upon? about 0.6. They converge upon what the true allele frequency is. That's kind of like if you flipped a coin, the more and more you flipped it, the bigger the sample size, the more you would converge on 50-50. So that's just 3 runs of that model. And here's kind of the message, is that genetic drift is probably not very important all by itself in large populations. So the random effects get averaged out over the sample size if there's a large enough sample size. But it could be really important in small populations. For example, these founding Silene dioica seeds are a small sample of the large population of the plants, and so they're not very likely to be the same as the mainland, and they're also not very likely to be the same as each other. So that's what are called founder effects. See if I can write that. See if my finger writing is getting any better. Founder effects is when a new population gets established, it's established by drawing individuals or zygotes or seeds or whatever from this larger pool of potential genetic variation. Suppose there's 10 alleles at a locus, and I start a population with 3 individuals. It's not possible for those 3 individuals to have all 10 alleles. We're assuming they're diploid, right? Just because there's no chance of recreating the same kind of genetic variation when you're starting or founding this new population. What happens with genetic drift, founders effects, and other kinds of things within small populations is you tend to lose alleles to fixation just by chance. And as you lose alleles to the fixation of the alternative allele, heterozygosity goes down. So here's a couple examples. The Silene dioica in these young populations, they're founder populations. In humans, there's this rare form of dwarfism that is, the alleles are about 1 in 1,000 in the world, but in the Pennsylvania Amish, it's at about 7% is that allele. So it's a huge, statistically a huge departure from what you would expect of any random population in the world, and that's due to the fact that that population is descended from a small number of individuals, 1 or a few of them, who happen to have this allele. And it's just increased in frequency in that population, not due to selection or anything like that, but just because it was a larger part of the genetic variation when that population started, and it's persisted over time. So here's what happens evolutionarily. In small populations over many generations, you lose allelic variation just by chance. Some alleles drift to fixation and other alleles drift to 0 frequency. Or if you have 10 alleles, maybe none of them are fixed, but some of them are being lost. Some of them are going to 0. And as that happens, heterozygosity goes down, because heterozygosity is the frequency of heterozygotes in the population. It's 2pq, or if you have lots of alleles, it's the sum of those, the frequency of those heterozygous genotypes. And in the book, they do this as 2p times 1 minus p, but you're all savvy enough to know that 1 minus p is the same as q if there's only 2 alleles. So what you see is that the frequency of heterozygotes is a positive function of the frequency of an allele up to 50%, and then it declines again. So if both alleles are present and at equal frequency, that's when you have highest heterozygosity. Now, obviously, if you have more than 2 alleles at a particular locus, you get highest heterozygosity when all alleles are present at equal frequencies. So when the A1 allele goes to fixation, heterozygosity goes down to 0. Or when it gets lost and the alternative allele goes to fixation, then heterozygosity is also 0. So here are outputs of the same kind of simulation model of showing genetic drift versus sample size over a whole bunch of generations. That's the left panel. And then the right panel show what happens to the heterozygosity in each of those populations. So here's a population of size 4, and the frequency of the A1 allele, obviously it bounces up and down randomly. It either within 20 generations, it either goes to fixation here or goes to 0, and the alternative allele goes to fixation, just by random effects. So this is the graph of average heterozygosity that goes with population size number 4. Heterozygosity within 20 generations or so, goes to 0 because you've lost that allelic diversity. Here's a population, exactly the same model, but it's an order of magnitude larger. Population size of 40 out to 100 generations. In several of those, you got alleles lost to going to 0 and other alleles being fixed. But in several, 1, 2, 3, in 4 of the runs of the model, both alleles persisted over the entire 150 generations, but you still have this declining heterozygosity as you lose alleles. Here's order of magnitude bigger, 400. You don't lose any alleles in that particular simulation model. But look, the allele frequencies change over time. They're bouncing up and down just as random effects, and there's this relatively slow erosion of heterozygosity. So it's illustrating heterozygosity as well as this really important effect of sample size. So you would like a way to understand what's the likelihood of these events happening, which is kind of a strange thing to say because genetic drift is an inherently random process. So you're asking to have a way of predicting a random kind of process, which you cannot do in any one instance. But if you were to rerun an evolutionary scenario many thousands of times, you could predict the average outcome. So here's the likelihood of fixation or loss of alleles just due to drift. And again, in any one instance, you have no predictive power. But in aggregate, over many such instances, you can predict that the probability that an allele goes to fixation is equal to its initial frequency in the population. So if there's some new mutation that makes some new allele that's selectively neutral, it's not advantageous or disadvantageous, it's not under selection, it's being just controlled by drift, a new mutation has a frequency of 1% in the population. What's the likelihood that it'll go from 1% to 100%? It's only 1%. You can see if there are going to be random changes in the frequency of that allele, it's much more likely to randomly change from 1% back to 0 than it is all the way up to 100%. Similarly, if you could view that as the alternate original allele that is now at frequency 99%, has a 99% chance of going up to fixation and this new mutant allele being lost. This is under drift alone. Can't emphasize that enough. If there was selection involved, it would be a completely different story. If the allele was advantageous or disadvantageous, completely change the dynamics. But here's a way that you can predict in aggregate what's the likelihood of an allele going to fixation or loss just by the accumulation of random events over lots of generations. And that's what it is. So here's an example by this guy Burry, who did an experiment with Drosophila, and he started the population with only heterozygotes, so he started with known P equals Q equals 0.5. The heterozygosity was 50%, so he started with that at the highest level. But what he did this with small populations, 8 females and 8 males. Then he replicated the experiment 107 times. So he had 107 different samples of a population of 16 individuals, and this is his average results, is that heterozygosity, as you would expect, goes down because of loss of alleles in these populations. So that, in a sense, matches your prediction, right? These small populations, you're going to lose alleles just through drift. Heterozygosity should go down. What you're not seeing is that the quantitative prediction isn't met of exactly how that heterozygosity should decay based on those size populations. So this little dashed gray line that's kind of hard to see, that's the quantitative prediction for a population of size 16. That's what you would expect heterozygosity to do. And his populations do lose heterozygosity like you would predict, but they lost it at a greater rate than your quantitative prediction suggests ought to happen. And again, it's replicated over 107 different lines of the experiment. So it wasn't just that, oh, one time it happened that the heterozygosity decayed more than you would expect. So his actual results, averaged across these 107 lines, are what you would expect if the population size was 9 instead of 16. And that brings up this idea of what's called effective population size. So when we look at count populations of organisms, we're usually thinking, again, at the organismal level instead of the genetic level. And for lots of the stuff we're talking about, you have to think at the genetic level. So how many individuals are actually breeding and contributing alleles to that gene pool in that population? That's the relevant thing, not how many individual organisms are there, how many are actually present. So if some of them died or they didn't mate, they didn't reproduce for whatever reason, then genetically the population size is smaller than the organismal population size. So this is the equation of the effective population size. N sub E is effective, sort of genetically effective population size. And what it is 4 times the number of males that actually reproduce times the number of females that actually reproduce divided by the sum of reproducing males and reproducing females. So it's completely independent of the non-reproducing organisms. It's only relevant those that are contributing genes to the gene pool of the next generation. So what you can see is if you have 5 males and 5 females that are all reproducing, equal sex ratio, that are all contribute kids, the effective population size is 10, which is the same as the organismal population size. But suppose one male mates with 9 females. Your population of organisms is still 10. But genetically, all of those next generation are descended from the same male. So the genetic variation that they have is reduced, right? They're all half siblings at least. So the effective population size is only 3.6. And this becomes relevant in lots of organisms that have polygynous mating systems where a large number or a large proportion of males don't reproduce offspring. The population genetically has less variation that it carries forward each generation because the offspring are sired by a few males rather than a whole lot. Okay. The other thing, so I guess the take-home message for most of what you're interested in terms of evolution and adaptation is that genetic drift is not important in large populations. But the caveat to that is if you have complete selective neutrality where alleles are equivalent to each other in fitness. There's no fitness penalty or benefit to these alleles. Their fate is controlled entirely by genetic drift. the likelihood of an allele going to fixation is equal to its initial frequency in the population. If you have all these selectively neutral alleles, if they constitute a large proportion of the genome of organisms, then you can do some interesting things with the molecular clock and tracking the average divergence rate between populations, things like that. So the idea of drift and the neutral theory, selectively neutral, becomes important in phylogenetics and the molecular clock and a lot of molecular genetics, whereas it's not, drift is not usually an important explanation for, say, differences among populations, provided they're large outbred populations that are not the result of founder effects or genetic bottlenecks. Right? So the molecular clock idea that we talked about before, the idea that that progresses at a, on average, constant rate is predicated on the genetic differences being selectively neutral. If they were not selectively neutral, then the differences would accumulate in response to the strength of selection. How divergent the selection forces is not in proportion to time. So for certain kinds of molecules, a molecular clock and genetic drift are going to be really accurate descriptions of how they evolve. For example, lots of third codon positions are synonymous, have synonymous substitutions. They have no fitness effect, and so that they diverge among populations roughly in proportion to time rather than any kind of selective agent. But would not be true for alleles that are selectively advantageous or disadvantageous. So, summary of genetic drift. These are random effects, random sampling error. Could happen in the example, it was the combination of gametes into zygotes, but it could be at any life stage. You know, if a tree gets hit by lightning and falls on your head, it's going to take out your alleles as well as your organismal fitness. If those alleles, if that random event gets rid of those alleles, if they're a random subset of the whole population, then it has no effect on the allelic composition of the population, because those kind of random bad events happen in proportion to the frequency of the alleles in the population, if provided you have a sufficient statistical sample size. Small population sizes are very much subject to genetic drift. They lose alleles, lose heterozygosity, and you always have to bear in mind that populations are genetically smaller than you think they are in a genetic sense. You never have effective population sizes that are larger than the organismal population size. They're always equal or smaller, right? Last thing of our 5 Hardy-Weinberg effects is non-random mating. And again, you have to go back to the genetic level, not the organismal level. At the organismal level, you don't mate randomly, right? You might end up with a telephone pole as a partner if you mated randomly. Mating with respect to particular alleles. Do certain alleles, are they more likely to pair up with each other or less likely? If they're in any way pairing up in frequencies that departs from that random expectation, then you have non-random mating with respect to those alleles, and that changes the genotype frequencies that you get, not necessarily changing the allele frequencies. We'll talk about sexual selection, which is non-random mating with respect to organismal characteristics in a couple weeks, I guess, whatever that is. So there's two kinds of non-random mating with respect to alleles. You could have positive assortative mating, where similar allelic states are found with each other more than you'd expect by chance, or negative assortative mating, where they're found with each other less likely than you'd expect by chance. Positive assortative mating is more common in most groups of organisms because it's what happens if there's inbreeding. So when genetic relatives mate with each other, they're both have inherited alleles from their common shared ancestors, so those alleles are more similar than the population as a whole. The opposite effect of negative assortative mating, when you're more likely to mate with someone of a genotype different than yours, apparently occurs with respect to immune system alleles. So when people check each other out and they're dating or whatever, they sniff each other, and some of the information you get about your partners is through sense of smell and how good, sexy smell. What that indicates is that their immune system alleles are different than yours, which would produce offspring with high heterozygosity of immune system alleles. So it's probably an adaptive smelling of your partners, right? Sniffing them out. But generally, positive assortative mating, inbreeding kinds of effects. And the consequence of this, in terms of population genetics, is that it lowers heterozygosity. You get fewer heterozygotes than you would expect from Hardy-Weinberg conditions. In other words, the heterozygotes are less than 2pq. So P and Q can change, the allele frequencies can move up and down, but you would still expect there to be 2pq heterozygotes. Whatever P and Q are numerically, you still expect 2pq heterozygotes. Finding fewer than 2pq heterozygotes suggests that something's going on. Maybe it's under dominance and they're being selected against. Maybe it's inbreeding by the production of fewer of them. So if you are picturing the genetic effects of inbreeding, imagine the extreme form, which would be self-fertilization, which would mean that homozygotes would produce only homozygous offspring, but heterozygotes would produce half homozygous offspring and half heterozygotes, right? The heterozygotes have P and Q. Well, they don't have P and Q. They have alternate alleles, A1, A2. So they would produce 50% heterozygotes, but also 50% homozygotes. Those homozygotes in the next generation produce only homozygotes. You never go backwards. You always decrease the frequency of heterozygotes in the population. And what happens is, per generation, the proportion of heterozygotes is expected to decrease by 50%. Without any change in allele frequency, you're just having fewer and fewer heterozygotes produced. One generation of random mating would restore your genotype frequencies to Hardy-Weinberg proportions. You haven't deviated from allele frequencies at all. So here's a table that just shows the frequency of heterozygotes per generation going down by 50% each generation. But just picture what would happen if at any one of those generations there was complete random mating. You would be back to 2pq expected heterozygotes. Do you all see that? You're like, what? I was sleeping. What? All right, well, hopefully. So, well, I just said this. If you sample a population, find more homozygotes than expected, could be inbreeding, could be something else. For example, maybe homozygotes have higher fitness than heterozygotes or whatever. But random mating restores those genotype frequencies. So, other effects of inbreeding that are important in terms of population genetics is that inbreeding increases the likelihood of an expression of deleterious recessives in homozygous state. If inbreeding increases the frequency of homozygotes, reduces the frequency of heterozygotes, increasing the frequency of homozygotes is also going to increase the expression of these deleterious recessive alleles into organisms that have lower fitness. That loss of fitness is what's called inbreeding depression. It's not the loss of fitness per individual, it's the average loss of fitness across that population due to the expression of these deleterious recessives. So here's a graph of the mortality rate among children of genetically unrelated parents here, plotted versus the mortality rate among children of first cousins, plotted here. And each point is a population or culture or some different particular human group. If there was no effect, if it was the same, then you would expect them to be equal, which would be on this line with a slope of 1, right? They're all or almost all above that line, meaning that the mortality rate with inbred parents is higher than the mortality rate with non-inbred parents. So that population-wide loss of fitness is called inbreeding depression and is noticeable in these human groups. It's also noticeable in wild populations of plants and animals. You can see a reduction in the average fitness of these different organisms based on this past history of inbreeding. So, there's this cool example in the book about conservation genetics that ties together several of these different kinds of evolutionary genetic effects that we've been talking about. This greater prairie chicken in Illinois. Oh, see if I can do this right. Not very likely. Okay, the prairie chicken. This might actually work. This is the first time I've ever tried this. So, that's a male prairie chicken doing his mating display. And they gather on these breeding grounds at these lek, what are called leks, or display sites. The male has this fancy orange crown, and he blows up air sacs and makes noises and stamps his feet and stuff like that. Very conspicuous, kind of sexy display. The females that you can see in the back, they're brown, cryptically colored. They're blend into the environment. They show up at these lek display grounds to check out the guys and find a mating partner. After they mate, the females fly off and raise the eggs and the offspring all by themselves. The males don't do anything for the kids. They don't make nests, they don't guard territories, they don't feed the kids, nothing. The females do all of the parenting. There's 2 different males in the screen now. So what happens in these leks typically is that females are fairly unanimous in their assessment of who's a sexy male and who they mate with versus who's not so sexy. So one or a few males get lots of mates and a whole bunch of males are losers. Some of you can relate to that. The sex, not the loser part, of course. Yeah. She's like, look at that one. Yeah, we'll talk about female choice and sexual selection and why females assess certain of the types of traits that they do later. But yeah, females are making an active choice. All right, so anyway, these things are endangered. In the early 19th century, there were millions of them, and then the steel plow was invented and introduced, and the prairie habitat changed completely because people planted wheat and corn and whatever stuff like that. And so from millions of these birds down to 25,000 in 1933, 2,000 in 1962, like 500 in 1972, down to, several tens or dozens by the 90s. These are in Illinois. So the reason why, of course, is this habitat loss for farming. So these maps show obviously dramatic change in habitat use, and these two remaining little habitat patches were protected in 1962 and 1967 as little reserves for these birds to breed. There they are. They're isolated from each other. Birds can't go from this one to that one. They're separate, isolated little pockets of populations. Here's a graph showing so these first one of these was protected in 1962, so these are data from 1963 onward. And what you see at first through the early 1970s is populations went up. And you think, wow, this is fantastic. We protected their habitat, and guess what? They go from roughly 50 individuals to 200 individuals, a 4 times increase in population in a few years. But the populations after that declined despite all these protections. So from the 70s through the 80s through the middle 90s, you are losing not losing populations, you're losing individuals within these two populations. They're shrinking and doing really poorly. And then in the middle-ish 1990s, the game managers decided to move birds in between these populations. They decided to impose gene flow, right? They swapped birds around so that they could breed with individuals from the other populations and bring in different genetics of the other groups, rather than staying in these small isolated populations that are inbred. They're breeding only within their small subgroup. So thinking in the context of stuff we talked about today, migration, genetic drift, inbreeding, you can look at the genetics of these populations that are threatened in terms of this kind of genetic stuff that we talked about. Allelic diversity was measured at microsatellite loci, and what you can see, here's 6 different loci from Illinois, Kansas, Minnesota, Nebraska. And then what's cool is in Illinois, they were able to measure genetic variation in populations before the decline using museum skin specimens, birds that were preserved that had still the genetic variation that existed before. And so what you can see is the number of alleles per locus in these different populations, again illustrating the idea that 2 alleles at a locus is pretty silly, right? The average in Illinois was 3.67. It was 5 or 6 in these other populations that were larger, more outbred populations. In Illinois, before the decline, the average number of alleles per locus was 5.12. So from 5.12 to 3.67 is showing you the loss of alleles at these loci over this period of decreasing population. So you have genetic drift. These are neutral loci. Genetic drift causing some alleles to go to fixation. Heterozygosity goes down. You lose genetic variation. And you have these small populations that are inbred. The effective population sizes are small, or the real population sizes are small, but the effective population sizes are even smaller because these are lek-breeding birds with these particular kinds of breeding things. So here's the percent eggs hatched from 1963 to 1990 has this generally downward trend. That's due to inbreeding depression. The loss of egg viability through inbreeding is an inbreeding depression effect. And look what happens after they start sort of forcibly outbreeding these birds, bringing in different genetic variation and getting rid of this inbreeding effect. The egg viability goes right back up. So the conservation genetics and the strategies that the reserve managers use is to reverse inbreeding by moving birds around. In a genetic sense, they're causing genetic drift to reverse this inbreeding effects and this loss of egg viability. Cool stuff. That's just a summary slide. Genetic drift, small populations, because of their lek mating system, their populations are even smaller than you would expect. They lose allelic diversity, they have low heterozygosity, and this management strategy of breeding new birds causes gene flow